Thank you very much. It's very exciting to be here today, and it's very fitting. Tomorrow we'll be celebrating Boeing's 100-year anniversary, and so that has very important ramifications for Genes in Space. Genes in Space was set up to encourage young men and women to continue their education in science, technology, engineering, and math. So we think Genes in Space is a perfect way to do that. And so we're looking forward to what happened, and we're looking forward to our continued journey this next year. So Genes in Space is set up to challenge students in grades 7 through 12 to design an experiment that will solve a space exploration problem or help solve one, and using DNA analysis. So the top 5 teams are selected. So, and those teams, and it's a very unique way we do the Genes in Space because we have mentors from Harvard and MIT for about a 6-week period that will mentor one-on-one with the 5 top teams. I'm not sure there's any other contest that's structured in that manner. So it's a very good way to do it. Let me see if I can change this here. Okay, I wanted to talk briefly on just kind of the history of Genes in Space and how it came about. So Boeing discovered a small device, a DNA thermal cycler, while we were participating in the mass challenge with our CASIS. So each year, Boeing and CASIS team up and we look at, go to mass challenge, and we look at all the various companies that have products at that event to see if there's any hardware or any companies that could take advantage of the unique environment of the space station. So in doing so, we did discover the mini PCR, and it was a situation where their product was already pretty well developed, so they didn't really need us in order to bring their product to market faster, but it was such a compact device, we realized there was nothing already on board the ISS that could do this type of biotechnology experiment. So it was a perfect fit. So we bought a couple of those devices, and that was in July of 2014, and for the next couple of months, we had some dialogue back and forth with the founders of the company. And we realized that the company was really focused on STEM education. Their goal was to bring that type of technology into the various middle schools and high schools across the country. So with that, we thought, how could we take advantage of that and put it onto the space station so that we could do something similar for the students. So we rolled at the National Science Teachers Association meeting in Chicago of 2015. In March, we rolled out the first Genes in Space. It was a very compressed schedule. We ended up rolling it out in March. The students had about 6 weeks that they could submit their proposals, so they didn't have a lot of time with all the other testing activities that were going on. So, but the results were very strong. We were really pleased. We got over 330 submissions. We were able to reach out to over 700 students in that 6-week period, and we represented 22 states across the country. So this year, when we rolled it out, we weren't sure what to expect, but we ended up growing the contest by about 28%. So we had over 900 students represented. We had over 400 teams submitted proposals, and we reached out to 26 states. So contests like this aren't possible unless you have a strong partnership. What we found is, of course, Boeing brought the expertise of being able to integrate the payloads onto the space station. CASIS was our partner in order to give us a flight opportunity. Mini PCR was the subject matter experts for the hardware that we used. Math for America was very key in being able to reach out to the different teachers across the country. New England Biolabs was instrumental for providing us resources to use their laboratories, reagents. You'll hear from Sofia, last year's winner, later on. She used their labs for their post-flight analysis. So this partnership was very strong, but we each bring something very unique to the table, and the contest could not have been possible without everyone's participation. So last year, this has been a busy year so far. In April, we flew the first sample. Anna Sofia's sample flew in April, and that was a very exciting time for all of us. At the same time, in parallel, we kicked off the second year of the Genes in Space. So we've been very busy. In May, we got the samples returned, and Anna Sofia, 17-year-old from New York, she'll explain the results to that later, so we were quite excited. But going through the integration process, there's a lot of change going on the ISS program right now, and it's really being focused on the payload users. That's the ultimate reason we have the space station. So we've been starting to allow the use of commercial off-the-shelf equipment, and we had not internally to Boeing taken advantage of that. So this was one of the first times we were able to take a piece of COTS hardware, integrate it on station, and then go back and look at our processes and be able to see what lessons did we learn from that experience that we might be able to influence our internal processes and make it easier for the other users. So a couple of key things that we learned: not all COTS hardware is it's created equal, not at all. Even though it's UL listed, there's different versions of being UL listed. So we had planned to fly a microcentrifuge. It ended up, when we did the testing on the centrifuge, it ended up being putting out the most electrical noise that had ever been tested for a piece of equipment on station. So obviously, we did not continue that effort. So the other thing we learned, when you're dealing with biological samples, I encourage you to go up front very early as you possibly can, to talk to your JSC toxicologist. Coordinate that well. That was imperative. That was one of the key roles that New England Biolabs assisted us with. So you need to be able to talk up front and use the proper language to make sure you're truly communicating. So we brought in a subject matter expert, our local toxicologist from Boeing, along with New England Biolabs. And once that really scientific, detailed chemistry discussion was taking place, things went pretty quick after that. Another one that's somewhat surprising that we discovered was in the area of cold stowage. So we needed to fly our samples at minus 20 degrees. So we had the cryo-approved NASA bags that we could use, the Ziploc bags. We had the cryo labels that we could put on them. We shipped all our hardware, got down to KSC, ready to get everything late stowed, and opened the bag, and all the labels had fallen on. So the adhesive on the cryo labels do not stick to the cryo bags. So lesson learned there: put some type of insert into the bag with your label on it, and then you'll be in good shape. So we were in a panic mode for a few hours to get everything relabeled. The other thing I learned was the crew members on board, they take a real interest in doing your science for you. They went above and beyond to get everything completed. They went the extra mile. They were very good to respond to every request we had. So overall, I think it was an extremely enjoyable experience for everyone. So later today, I think at 12:15, I hope most of you can be here, we will be announcing the winner for the 2015 or 2016 Genes in Space, the number 2. And we're very excited to that. And I think next, I'd like to have Zeke come in. He's our colleague from Mini PCR, and he's the founder and the designer of the device. So he'll talk about what the polymerase chain reaction really is and a little bit about the hardware. Thank you.
Thanks, Scott. Good morning, everyone. Okay, the big one with the star. Tech support. That's the pointer. I don't see a big. So, good morning, everyone. Many of you may be wondering exactly what Genes in Space is and what kind of projects the students are proposing. So I'd like to take a few minutes to explain this science a bit more in detail to you. So when we look at the stars, we use tools. We use a microscope, a telescope. And the same happens when we have to look at DNA. We need special tools. DNA is very small, so the equivalent of the telescope for DNA is something called PCR. So for example, when an astronaut might swab a surface of the space station to look for the presence of bacteria, or they might even take a cheek swab to look at their own genes, how do they go from that sample to actually getting a result for what they want? Presence or absence of bacteria, for example. So they use something called PCR, the polymerase chain reaction. What PCR allows anyone to do is to focus in a very small region of DNA out of a very big region. For example, the human genome has about 3 billion letters. Through PCR, you can focus on a very small gene. And it allows us to make many, many millions of copies of DNA in a short amount of time. So once we have all these copies, we can then go and analyze it, either by sequencing or by directly looking at that DNA. It's an extremely powerful technology. We can detect a single molecule. And it's used by scientists and doctors daily, millions of times. So if anyone of you is going to Rio later this month for the Olympics, when you come back, you might want to be checked for a Zika virus, for example. So you'll ask your doctor to do that. He would do PCR to find that. PCR is also used to look at other viruses such as HIV, hepatitis C. It's used in the clinic all the time. If you're concerned about carrying a genetic mutation that might make you predisposed to developing a disease such as cancer, again, you would get a sample taken, someone would do PCR and look for a specific change in your DNA. So it's extremely important in medicine. We also use PCR in many different ways. For example, to look at our ancestry and how we relate to each other and to other species. We use it in forensics all the time to maybe implicate people in a crime or actually take some people out of jail. We use it in crop biotechnology to modify them to hopefully make them better. So DNA is really here to stay. So what we've done for Genes in Space, if we put one of these PCR machines that allows us to look at DNA, and this is what it looks like. It's very small. So we put this in the space station, and it's staying there for Genes in Space competitions and for use by other people too. So once we had it up there, we asked students, what will you do with a PCR machine in space? And we gave them a few examples, ideas to get started thinking. But what we find is that most of the ideas ended up in that last box that says your idea. These students are incredibly creative, and from those 380 submissions we got this second year, most of them ended up in that bucket. The students are very creative. And yesterday, we heard from 5 of these groups, and they talked to us about how genes may transfer between bacteria in bacterial biofilms in space and how they might make them more virulent, for example. Or antibiotic resistance. How we can look at the ends of the chromosomes, which are implicated in aging. How we can monitor oxidative stress in almost real time. How viruses that are latent in the astronaut's DNA might get reactivated. And finally, how mutations might behave differently on Earth than in space. These were all incredible ideas, and I couldn't help but notice that during the first day of the conference, all these 5 ideas were mentioned by the speakers, either by the Kelly brothers, and Sanjay Gupta, and Topol. So I think what that tells us, the grown-ups here, or the older kids, is that we ought to pay real attention to these kids. They're very sharp, they've done amazing work, and we should be proud of them and looking at them very carefully. So this picture is from astronaut Team Peak, actually performing the first PCR in space ever done. And in that picture, he's actually talking to Sofia Bulgaria. As Scott said, she was the winner of last year's competition. And it's incredible to think that the first molecular analysis of this kind was done for a high school student by an astronaut there. So without taking more time, I'd like to introduce Sofia to tell us her story, and she's going to share with us her results. Thank you.
Hello, I'm Sofia Bulgaria, and as mentioned, I was the winner of the Genes in Space competition last year. As part of this competition, I got to do an experiment that is the initial stages that will contribute to the development of a polymerase chain reaction, or a PCR-based assay, for evaluating epigenetic changes in the human immune system in space. Oh, look at that. Okay. So the human immune system is a network of cells, tissues, and organs that all work together to keep us healthy. And one problem that astronauts often have is a depression of the immune system. They suffer from many immune system defects. And if we hope to go into long-term spaceflight, and if we even want to keep astronauts on orbit, it's going to be very important that we understand how their immune systems are functioning and how they're not functioning. So, of the cell types in the immune system, the one I focused on were the T helper cells. T helper cells initially start as a naive CD4 plus precursor cell, and then depending on signals in the extracellular environment, either differentiate into Th1 or Th2 cells. So, these T helper cells are then identified by the cytokines or signaling molecules that they secrete, and they each secrete their own unique array of these molecules. Th1 cells promote cell-mediated immunity, or a T-cell-based reaction to aberrant major histocompatibility complex markers on other cells, and listed are some of the cytokines they express, including interferon gamma, interleukin-2, and tumor necrosis factor beta. Now, T helper 2 cells, on the other hand, promote humoral immunity, and this is a B-cell-based reaction to circulating antigens or pathogens in the bloodstream, and some of their cytokines include interleukin-4, interleukin-10, and interleukin-5. Now, it is well known that spaceflight impairs astronauts' overall immune response. Not only does it cause an immune system depression, but there's also evidence that while in space, there's a decrease in the ratio of interferon gamma to interleukin-10. This indicates a shift to a T helper 2 dominant phenotype in astronauts. Not only does a long-term T helper 2 shift cause a significant decrease in your cell-mediated immunity, but more specifically, it increases your risk of autoimmune diseases, your susceptibility to allergens, and your general susceptibility to other illnesses. So, another important part of this puzzle is epigenetics, or a layer of DNA modifications that's added over the genome. So you have your base sequence of DNA, but then you have markers that go over it that affect then how those genes are expressed. The markers that I cared about are called methylation markers, or DNA methylation. What they essentially are is at CG dinucleotides, so locations in the genome where C's and G's are next to each other, there's the addition occasionally of a methyl group to the cytosine residues in the DNA. And what this causes is a condensation of the DNA strand, which then prevents the transcription or the access to whatever DNA sequence is under these methyl markers. Now, this is often used in gene expression. It's a mechanism of gene expression in our body, and the way this is controlled is the promoter of a gene will be methylated to silence the transcription of that gene. Now, DNA methylation is very important for Th cell differentiation. If you remember the cytokines I discussed earlier, they all have various promoter regions, and the methylation or demethylation of these promoter regions controls their differentiation into specific cell types. So as you can see, this is CNS6 and then the interleukin-4 promoter. This occurs in almost all of the cytokines. I just highlighted these two as the top is specific to Th1 cells, the bottom specific to Th2. And as you see, as they differentiate into the different T helper cell types, the methylation pattern at the promoter changes, which then changes the expression pattern of these cytokines, allowing the cells to have their specific expression pattern. Now, what's probably most important to bring this puzzle together is that previous research has indicated that spaceflight causes changes in astronauts' methylation profiles. This is due to not only physical stressors such as cosmic radiation and the presence of microgravity, or I guess the absence of gravity, but also just the sheer stress of being in a spaceflight environment, the stress of launch, somewhere where you have to be in this closed space where you have to be on top of things at every second. All of these can change the human methylation profile. So I thought it would be very important to figure out a way to evaluate this methylation profile, to look at how astronauts' methylation profiles are doing, see if there's a connection between the methylation profiles of their immune genes and the immune system defects they're experiencing. And if I was able to characterize this, then looking at the development of, as I mentioned before, a PCR-based assay for evaluating these changes in space and possibly tracking them. So we have some epigenetic warning before we see a phenotypic change in the astronauts. We may be able to do something to mitigate the change. Now, to do this, I wanted to use a technique known as bisulfite PCR. What this entails is the treatment of a sequence of DNA with sodium bisulfite, which changes all unmethylated cytosines to uracils. This means that you have a different DNA sequence depending on whether or not you had methylation. And then you can use techniques such as methylation-specific PCR to determine kind of what % methylation you had and how your genome was methylated, because you can target different sequences depending on whether or not your genes are methylated. But before I could do any of this, I had to make sure, can we amplify DNA in space? PCR had never been performed on the space station, so I thought it was probably best to start with a proof of concept to make sure that we weren't just going out there and using a technique that hadn't been validated. While I was doing that, I also wanted to check if we could amplify bisulfite-converted DNA in space. The significance of this being a bisulfite treatment significantly increases the fragility of the DNA, so it's important to make sure that that is still able to be amplified in the space environment. And then I wanted to know, can we use these techniques to assay immune-related genes? This is going back then to looking at the immune system and understanding astronauts' immune system. So I ended up running 2 samples in parallel with genes from zebrafish, and the DNA we used zebrafish DNA because the methylation profile of during development, the methylation profile of the zebrafish is very well characterized, and for this we wanted something that we knew exactly what we should be seeing. So the samples were prepared on the ground. We had 2 sets of samples. Then one of these sets was actually sent to space. What was sent to space was a complete mixture of everything you need for PCR. We had the DNA, we had the primers, we had the polymerase. Everything was already enclosed in the tube. All the astronauts had to do was take it and place it in this PCR machine and run the protocol that we had given them. While they were doing that, we did the exact same thing on parallel in Earth, with the hopes that when we got it returned, we would see the same amplification profiles, we would see the same thing that had occurred on Earth and space to confirm that this technique does work. Now, as you can see, the top row of samples are our space samples, and the bottom is our Earth control. This did come out slightly darker on these screens, so it's not as easy to see. But the nice thing we saw is, though there was you can see some residual kind of primers hanging around, you can see it's not as clean as maybe an earthbound PCR experiment would end up when you ran it out on a gel. But I think this is probably due to the fact that this did sit for a very long time, and then this did suffer the stresses of both entering and exiting space. We know that there's no issue with the polymerases thanks to the help of the people at New England Biolabs who did a bunch of testing on them for us to make sure that they could sit and make sure that they would withstand just being held around in cold storage, waiting for their turn to go on the space station. But anyway, so if you see along the top, you have CM and CU, that's designating converted methylated. So those are the primers which would amplify if there was methylation, and then, CM, and then CU is converted unmethylated. So those are the primers that would amplify if there wasn't methylation. And then we also have a set of unconverted samples. And then 0 hour and 6 hour just indicate the time point when we extracted the DNA. The first gene we looked at was interleukin-4, which is one of the immune genes I was discussing earlier. And we looked at a region within its promoter where we understood what the methylation profile should look like. And as you can see, there's a really nice amount of similarities between what we saw on Earth and what we saw in space. This answers for us all three of our questions. Can we amplify DNA in space? Yes, those bands in the space sample, that's DNA. That's being amplified on the space station and come back. This process works. And then, can we amplify bisulfite converted DNA in space? Again, yes. Remember, the C's designate converted. Those are the converted samples. And finally, can we assay immune-related genes? Yes, interleukin-4 is one of these cytokines. It's one of the cytokines I discussed right at the beginning of my presentation. So this confirms that this technique works. Of course, because this is a preliminary experiment, just a proof of concept, there's much more optimization that can go into improving the quality of these assays, making sure that we can get cleaner and cleaner results. But that's innovation that's now open to everyone. Not only the Next Genes in Space winner will go on to improve the quality of PCR in space. They'll be doing their experiment. But also, anyone else who sees a use for this PCR machine in space will be able to work to ensure that they've got the best quality experiment they can have. I also did look at HOXB3A, which is just a developmental gene. And again, we looked at this just to see it's a well-characterized gene, and we wanted it in case the immune gene didn't work for some reason to fall back on. But essentially, what we learned is yes, this little box does do what it's meant to do. We now have the capability, thanks to mini-PCR, to amplify DNA in space, which is something we had never previously been able to do. Now, I owe a lot of people thank yous for letting me do this research. I want to thank, first and foremost, mini-PCR for coming up with the box and starting this competition. I'd also like to thank everyone at Boeing and CASIS who helped me get my experiment flying. As you can see, that little white thing on the, that is the rocket, I assure you. So that's Boeing and CASIS, thank you to them. Also, a huge thank you to everyone at New England Biolabs, not only for their help establishing which polymerases we could use, but also for giving us lab space to work. We did the final post-experimental analysis at New England Biolabs. And then I'd also like to thank, I had a mentor who helped me, Holly Christensen from MIT. She was a huge help in this. And then thank you to my parents, and I'm sure I'm forgetting people, but thank you to everyone who helped me, because what they've enabled is not only that I can understand that this works, and this then opens up the potential to moving towards that assay I was talking about before, looking at astronauts' immune systems, but they've also opened up the possibility of looking at DNA, looking at PCR-amplified DNA in space to anyone who wants to use the space station for their future innovation. Thank you.